Hello and welcome to our fifth educational webinar, Genome Editing Chats, organized by and for young researchers, supported and sponsored by GenomeD, our cost action network focused on genome editing for the treatment of human diseases, and in collaboration with CRISPR Medicine News. My name is Laura Torella, and I'm the young researcher, investigator, innovators of GenomeD. And I'm delighted to welcome you today to our new webinars on a very hot topic in genome editing, which is delivery. And especially today, we will hear about advances and limitation of in vivo delivery systems for therapeutic genome editing. I have a great pleasure to have you, Nerea, as speakers for today's presentation. Dr. Nerea Zabaleta recently joined CIMA, University of Navarra, as a researcher. And the main focus of her research is the generation of novel gene delivery vehicles to enable the development of in vivo genome editing for inherited kidney diseases. Dr. Zabaleta performed her studies at the University of Navarra. She graduated in biochemistry in 2013, and then she also got a master in biochemical research in 2014. She then performed her PhD thesis in the laboratory of Dr. Gloria González Aceguino-Laza at CIMA between 2014 and 18. And she was working on the development of an in vivo gene editing approach for the treatment of primary hyperoxaluria, an inherited metabolic liver disease. In 2019, she started a postdoc, a fellowship in the laboratory of Duke Vanderberg at Grossbeck Gene Therapy Center at Harvard Medical School, which lasted for 3 years. And during her postdoc, she developed novel vaccines platform based on immunologic adeno-associated viral vector. And this experience led her to the promotion to a structure in 2022. As an independent researcher and first as an instructor between 2022 and 2023, and later as an assistant professor in 2024, Dr. Zabaleta studied the biology behind the biomanufacturing of AAV with the goal of improving the production of important clinical vector. And she also focused her efforts on capsid engineering to generate a hard-targeting AAV capsid. And after obtaining from Monica Hall grant, she got back to CIMA, where she relocated her lab. And she has 18 peer-reviewed publications, or as last author, and she is also the co-inventor in 4 patents. Nerea is member of our GenomeD Action, and she's active in the working group 2, and working group 3 and working group 5. We will look at her also for the working group 2. On top of everything, I may say that Nerea is an incredible mentor. She's a super nice and good partner in science, and she's the researcher to which you will reserve the most brilliant career. So, Nerea, I want to just remind the audience that we have a Q&A chat. So please type in all your questions, tabs that we will discuss with Nerea at the end of our session in the Q&A session. And now, Nerea, over to you. We are very much looking forward to hear about advances and limitation of in vivo viral delivery system for therapeutic genome editing. The floor is yours, and you have more or less 45 minutes. Thank you.
Thanks so much, Laura. Thank you for this great introduction. It's a real pleasure to be here hosted by you. And well, as you said, today I'm going to focus more on the delivery side of things, which is my main expertise. I work with gene editing, but mostly using tools that other people have developed. But our lab focuses a lot on development of new, especially viral delivery systems. So let's maybe start from the basics. I think this is important to remember. So in gene therapy, there are 3 very important factors. One of them is the target. So we have to define what is our disease target, what is our target organ, what is our target gene. Once we know that, we start thinking about the molecular intervention. So are we going to increase the expression of a gene? Are we going to silence the expression? Are we going to modify the gene? And how are we going to do all of that? And finally, and a very important part is the delivery. So how are we going to bring all these ideas and strategies to the target cell, both ex vivo or in vivo? So in this case, and I think for this audience, there is a very big focus on the molecular intervention, which is the CRISPR-mediated genome editing. So as you know, we have many different CRISPR approaches, many different Cas proteins, and many different strategies that we can do by using these tools. However, I think something important to remember is that the editing efficiency doesn't only depend on the specific tool and the specific design we are using, but it also depends on the efficacy of the delivery method. So if you have a great Cas that does a great prime editing, for example, that's perfect. But if you don't have a delivery method that takes that editing machinery to the target cell, you are not going to have any therapeutic efficacy. So this is a recently published review, and I found this table, which I think it's pretty interesting. These are the clinical trials that have been done with gene editing in vivo. And you can see here, if we look at the delivery vector, you can see that it's clearly dominated by two delivery methods. One of them is adeno-associated viruses, and we're going to talk a lot about them. And the other one is lipid nanoparticles. So this is great, and it seems to be a 50-50 differentiation between these two. But something also of interest, if you look at the disease target, you can see that all the trials with LNPs target the same organ. They target liver, while the trials using AAV target other organs like retina, immune system, skeletal muscle, and central nervous system. This is already showing us some trends with these different vectors, and it's also going to show us what are the advantages and the disadvantages of each of them. So let's dive a little bit into the LNPs. I'm going to just give a very brief overview of the LNPs because it's not, my field of expertise, but I just wanted to give you a little bit of an intro. So LNPs are, as the name said, lipid nanoparticles, and here what we do is conjugate different lipids with the mRNA, and in our case we would use an mRNA that expresses the Cas9 or the Cas, and an mRNA that is the guide RNA in RNA format. So these lipids have different modifications, and we also can modify them to enable tissue targeting. So we can decorate the lipid nanoparticles with molecules that will bind to receptors in the target cell, and they will provide an improved targeting to the target tissue. I'm going to give you an example, which I think most of you will already be familiar with, and this is the inhibition of transthyretin or TTR in hereditary transthyretin amyloidosis. So this is a disease in which there is a mutation in the TTR gene, and due to that mutation, the TTR tends to aggregate and create amyloid fibrils. So TTR is produced in the liver, it is secreted, and the mutated TTR will produce these amyloids that will be deposited in different tissues, causing a variety of symptoms in the patients. And this is autosomal dominant. So by having just one copy of the gene mutated, you are going to have disease. So this is a trial that was done with LNP CRISPR by the company Intellia. And here what they did was package in LNPs the mRNA of Streptococcus pyogenes Cas9 and the guide RNA that is specific for the TTR gene. These lipid nanoparticles were injected intravenously in the patient, so these are injected systemically, and they transfect, I guess, very well the liver. So the nanoparticle comes from blood, it's uptaken by the hepatocytes and by the endosomal route, and they escape endosome. Both products are, the Cas9 is translated and bound to the guide RNA, and this goes to the nucleus and generates a double-strand break in the TTR gene, following by indels. And this strategy is used to disrupt the gene and reduce its expression. So this is the clinical data. As you can see, the lower dose decreases more or less like a 50% of the TTR protein in circulation, and the high dose gets you to an 80 to 96%, which is an amazing number. So you can see that they are probably creating indels in most of the hepatocytes in the TTR gene. LNPs have certain, as everything, have certain advantages and disadvantages. So they are mostly non-immunogenic. They are efficiently encapsulated. They can give you a targeted delivery. So you can, as I said, you can decorate them to have a specific delivery, although this is not always efficient. They have a transient expression. So with this, you achieve a transient expression of the Cas. Probably it's around for like a week or so. And this is going to reduce the chances for off targets, because you won't have the Cas, in this case, the Cas9 expressed for longer and lying around there, finding different target sites. And the production is very scalable. So as you know, with all the experience with the COVID-19 vaccines, the production of LNPs was very well streamlined and very well optimized. However, there are certain issues, and some of them are not as much studied. For example, there is certain immune activation, and it is unknown how much toxicity that could give you. And I think one of the biggest challenges, especially if we are thinking about genetic diseases in general, is that targeting a non-liver tissue is very difficult. LNPs are very good and very promising, but I think that at this point we are having a lot of issues to achieve efficacy in other tissues. So that's why I think it's important at this point to look into other delivery methods, such as viral vectors. So let's talk about viral vectors. So how would you select a viral vector, right? Like we often don't think about it because we have them already given, so we know which viral vectors exist. And but I think it's important also to make this exercise of, if you were to choose a virus to vectorize it, which one would you choose? So there are certain things that you need to keep in mind. So for example, safety is an important one, right? Is the virus pathogenic? Which you can avoid, because for example we use lenti based on HIV, which is very pathogenic, but we can engineer it enough to make it safe as a vector. But some things are important, such as which elements of the virus remain in your vector, and are they gonna give you immunity? Immunity that is going to cause toxicity, but immunity that could also cause an immune reaction against your expressed transgene, and that's going to reduce your efficacy as well. Then there are some practical considerations. For example, are you able to produce a vector? Because it might be the best vector under the sun, but if you are not able to produce it at clinical grade, it's never going to get to anything. So can you produce it? What are the yields? Can you scale it up? Something very important, I think, for gene editing, what is the genetic cargo capacity? And we will talk about it in a few slides. And finally, I think, which is something we think most about, which is the efficacy. Is it potent enough? Has it a broad tropism? Is it able to give you a specific transduction? is there pre-existing immunity in humans? And this is something, for example, if you are injecting vectors intravenously and there are pre-existing neutralizing antibodies, the vector is going to be completely neutralized and it's never going to reach your target. And also thinking about the life cycle of a vector, it's very different of a life cycle of a virus, right? So you know that when we have a virus, it has its gene that has the terminal repeats, which are packaging signals, and it has all the viral genes. So this virus infects the cell, releases its genetic cargo there, and thanks to that, all other the viral proteins are produced, and these viral proteins start the replication cycle, and new virions are generated, which are then going to be released from the cell, and these virions will initiate again the infection process, and we will have more and more virus. In a vector, we change this a little bit. And the cycle is divided in two. The first one would be production. So we have our vector genome that has the packaging signals here, but in between them, instead of the viral genes, now we have our cassette of interest with a transgene, a promoter, whatever you want to put in there. And in trans, we will provide the viral genes that are going to help, like, for example, the replicase, the capsid genes, and we will transfect all these elements into cells. And when the viral genes are produced, the replication cycle will start and vector particles will be produced, which will be released, or we can just harvest them from the cell. But there won't be an amplification in principle with the vectors we want to use for therapy. There won't be an amplification. So we will purify this vector, and then we will do the second part of it, which will be what would be similar to an infection in the virus, but it would be the transduction. And in this case, the vector enters the cell, releases the genetic cargo, and this genetic cargo produces your protein of interest, or carries your gene of interest, and it's gonna be expressed in the cell, or it's gonna do the whatever function you want. So there are three main vectors that people have used clinically. We have retroviruses and lentiviruses. These are relatively large viruses with a genome of around 10 kb. The genome size is going to more or less tell you how much you can package within them, not always, but it produces a stable transgene expression, and they are able to integrate the transgene in the genome, although there are also some non-integrative versions. And they are mostly used ex vivo. However, for editing, they are not as used ex vivo because as I said, we don't want to have a long-term expression of the Cas, so other methods are used. And there are somehow used, in vivo, but not that much. Then we have adenovirus. This is also relatively large. It has a very large genome. And there are several generations of adenoviral vectors. The first generation has more viral genes, is more immunogenic, doesn't have as stable expression. But for example, the last generation are what are called the gutless adenoviruses, which are devoid of all the viral genes, and you can package there as much DNA as you want, almost like 30-something kilobases. And that can give you a longer expression. So they are not very commonly used, nor ex vivo nor in vivo for therapeutics, although they have, as you probably are familiar, they have been very extensively used in vaccine applications in vivo. And finally, we have the adeno-associated viruses, or AAV, which are very tiny viruses with a small genome, but they provide a very long-term expression because they remain episomal in the cells that they transduce. They are not very widely used ex vivo, although for editing they are used as a vector to deliver the homology-directed repair template, and they are very, very widely used in vivo. So I'm going to focus here because I think right now for in vivo gene editing, as you saw, it's the most used vector, or the only used vector, viral vector, to be honest. And I think that it has very important features. So AAV is a single-stranded DNA parvovirus. It's, I would say, almost a nanoparticle because it has a small genome that contains inverted terminal repeats, which are the packaging signals, and 2 genes. Two. Several others, but mostly rep and cap. And it's icosahedral capsid, and it's a non-enveloped virus, and this is just a protein. So it's a protein, a very short DNA. And this is replication incompetent. It needs a helper virus in order to propagate when it is a virus. As a vector, we just remove these viral genes. We only keep the ITRs. So there is no viral open reading frame in the vector. And we substitute them by the expression cassette. And we will use a rep and cap plasmid to produce the vector. So there are, I think, many advantages and many disadvantages. Something that I think is very key is that AAV has been around for a long time already. So I think early 2000s probably we started getting the first clinical data with AAV. So we are very aware of what the disadvantages are. So, so many patients have been treated with AAV that it's been very clear where toxicities are, what our limitations, how much AAV we can administer, and other issues. So the disadvantage list is very large, and I think it is because we know much more about them and how they behave clinically. The advantages are that it transduces very highly. So for example, if you do experiments in mouse, you could almost transduce every single tissue. It's relatively easy. It's different in human. They have different serotypes of AAV, and they have a broad tropism, but we can also tune the tropism. We can modify the AAV capsid in order to alter the tropism, and we will talk about it in the next slide. We have extensive clinical experience, so this gives AAV a head start over other vectors. There is established manufacturing, and it has a lower immunogenicity than other viral vectors, for example, adenovirus. However, some issues, and this is very important, and we will look at this also in one of the next slides. There is a limited packaging capacity, so this is a problem for CRISPR, where the Cas proteins the Cas genes are usually long. There is a long-term expression, which is an advantage for other applications, but not for CRISPR, because we don't want to have the CRISPR machinery expressing there for long, because it can have higher chances of target editing, as well as potential immune responses against the CRISPR system. So if we have the foreign protein there expressed for a long time, the immune system might pick up on that and generate a response against the CRISPR components. There is pre-existing immunity, so most of us have been infected by wild-type AAV, even though it's not pathogenic. Most of us have pre-existing immunity, and this is an issue because we develop neutralizing antibodies that can neutralize the vector as soon as we inject it, and it's never going to get to its target. And finally, viral vectors can integrate, although there is no active integration. Most of the vector genome remains episomal, but there is some integration that can cause genotoxicity. So let's talk about the AAV capsid. So I wanted to show you this slide, and this is a very busy slide because it contains a lot of work that has been done over many, many years. But AAV, you have to think about AAV as something that is many different vectors. AAV encompasses many different vectors because we have many different capsids that have very, very different profiles. So they have different tropism, they have a different immunogenicity, some of them different manufacturability. So, whenever you have an AAV capsid, you have to really study it separately of others. So there are different ways that the field have come up with to generate new AAV capsids. Biomining was the traditional way. So this was how people discovered the first AAVs. So they would get to, they would go to human or non-human primate tissue and PCR up the cap gene and see what they picked up. So that's how they defined all the what we call natural serotypes. So AAV9, for example, which is very widely used, AAV5, AAV8. There are many AAVs. If you see that they are RH dot something or HU dot something, they are also natural. So this is from rhesus macaque, from human. But then this was seen as somehow limited. So people came up with new ideas to modify capsids and to also screen for modified capsids. So, for example, unbiased mutagenesis. One of the strategies is capsid shuffling. So for example, you take many different natural serotypes, you chop them, and then you shuffle them, and you create wholly new capsid that have, it's like a Frankenstein of a capsid. It has different parts of different capsid, capsid sequences. Or you can do also error-prone PCR. What people do with this is they generate these huge libraries which have many, many different capsids, and then they screen them in a system that is going to allow them to select the capsid that works best in their system. Then there is rational design, and there is something that people do. It's in between rational design and directed evolution, which, not necessarily directed evolution as we understand it, for example, for CRISPR, but which is, we know that we can insert a peptide in a safe harbor in the capsid. And most of the time we use this insertion to direct AAV to bind to a specific receptor. So I think Mayo AAV is pretty popular, you might have heard of. So this Mayo AAV is an AAV9 that has a peptide inserted in a specific region of the capsid that is in the outside part of the capsid, and the peptide carries an RGD domain that binds to integrins. And with this, what we achieve is that AAV transduces muscle very, very efficiently. So this has been done, with other peptides, and there are several other vectors that were derived in this way. And then the pure rational design, there have been different strategies like sequence-based, structure-based, phylogenetic. We are now more and more using machine learning. One of the limitations of the rational design is that the understanding of the structure function, let's say, the structure tropism relationship is not as good. So we don't exactly understand why an AAV8 transduces preferentially liver in a mouse, but an AAV9 transduces liver plus heart plus CNS. So we don't exactly know what structural parts of the AAVs are providing that phenotype. I'm not going to enter more into AAV capsid engineering. This is a very big field, and part of my lab works on this. But I'm happy to take any questions at the end of the talk. Then let's talk about the size limitation, which is, I think it's a big one and an important one. I really liked this picture from this review. Here you have the different elements that you can add to a AAV for a CRISPR approach. So you have the guide RNA, donor template, not as frequent for in vivo, for example. We have the Pol2 polymerase 2 promoters to drive the expression of the Cas, Pol3 promoters to drive the expression of the guide RNA, poly-A for the Cas. We may have inteins as well sometimes. And then we have, which is the biggest problem, the editor. So as you see, we have some editors that are small, but the editors that we have used traditionally, like SPCas9, is very large, is 4.2 kilobases, and the cargo capacity of AAV is 4.7. This 4.7 includes ITRs, promoters, poly signals, the editor, everything. So very early on, the Staphylococcus aureus Cas9 was described as one of the promising editors for in vivo, and it has been used widely, and I will show you a clinical example with this Cas9 because it fits an AAV and it gives you some flexibility for promoters. Something important to note is that the longer the promoter, the easier it's going to be for you to achieve a specificity of expression. And this is also very important to consider in terms of toxicity. So, we have short promoters. For example, the EFS promoter is very short, but it's ubiquitous, and it's going to give you expression everywhere. You might want to have a longer promoter and a more specific expression. And then we have the base editors and prime editors, which are huge because they have extra sequence, right? Like they have the base editor fused to the Cas9, or the retrotranscriptase fused to the Cas9. So these directly don't even fit into an AAV, even if you don't have any regulatory elements. So people have come up with different strategies to deliver the editors using AAV. So you can have an all-in-one AAV that has the editor plus the guide RNA plus everything you need. This can be done with only some editors. You have dual AAV with or without split editors. So here you may be able to fit the editor in one AAV, but you need an extra AAV for other elements like the guide RNA or the donor. And in the split editor AAVs, people traditionally use inteins to have part of the editor in one vector and part in the other, and after the translation they reconstitute the protein in the cell. So this is an example in vivo. I think it's the only clinical trial with AAV and CRISPR, if I'm not wrong, that has published clinical data. And this is for a retinal disorder. It's leber congenital amaurosis caused by mutations in CEP290 gene. And this is the approach they use. They use an aureus Cas9, the one I told you that is smaller, on the smaller end. And they have two guide RNAs, everything in a single vector. And they use an AAV5 here. So this is the mutation they are targeting is a splice variant. So there is this mutation in this intron that causes a disruption in the splicing, and therefore the protein is not properly produced. So they are targeting the intron itself with these two guide RNAs, aiming at deleting this region or at causing an inversion that is gonna restore the proper splicing. So here I'm just putting, you can read the paper, is this one. I'm just putting a piece of data on therapeutic efficacy. They have treated 14 patients, and they see that in 6 out of 14 they have a positive clinical outcome. Okay, so to this point I've explained more or less a little bit of the how the field is and a little bit of the theoretical part. And now I'm going to start with a case study that was done by us. And it's going to allow you to see a little bit more in the, the little details on how you can run an in vivo gene editing project. So we're going to talk about primary hyperoxaluria. This is an autosomal recessive disease caused by mutations in the AXT gene. And I see that I'm almost running out of time, but I'll try to be fast. Mutations in this gene that cause disruption in the AGT protein that is important in the glyoxylate metabolism in the liver. So when we have this mutation, glyoxylate gets accumulated and it's metabolized into oxalate that is very toxic for kidneys because it deposits in crystals, causing end-stage renal disease. And this is the onset is mostly in childhood, so it's a pretty difficult disease for children and parents. There is a therapy that is based on an interference RNA, where the strategy is a substrate reduction therapy. So basically what they do is inhibit this enzyme here, glycolate oxidase or GO, which generates the glyoxylate. So if you inhibit GO, what happens is that glyoxylate is reduced and therefore oxalate is reduced, and you will eliminate that overproduction of oxalate that will cause the kidney damage. So therefore we reduce oxalate by but we increase the glycolate here, because if we eliminate GO, glycolate gets accumulated. However, it has been published that some people have mutations in 2 copies of the HAO1 gene, which is the gene that produces GO, and they have an asymptomatic elevation of urine glycolate. So it seems like knocking out GO, it's not a big deal for the physiology. Based on that, this is an Alnylam product, which is called Oxlumo or lumasiran. And it has been shown to reduce oxalate both in urine and plasma, depending on the stage of the disease. However, it has some disadvantages, which are multiple administrations are needed. So there is sometimes an issue with therapy compliance, and long-term effectiveness is still unknown. So we decided to generate a therapy based on CRISPR to disrupt the HAO1 gene, and all of the data I'm going to show you has been generated in mouse. So here we use an AAV8 vector, which I've said before is very good to transduce the liver of mice. And we designed 2 guide RNAs that target the exon 2 of the HAO1 gene in the mouse. So the goal is to express the Cas9 in the liver together with 1 guide RNA, and this is going to create a double-strand break and the indel generation through non-homologous enjoining. After this, we also tested other 2 strategies using the same guides all the time. So we combined the nuclease with both guides. So we will have 2 double-strand breaks that will result in, as defined before, in a precise editing. So this cuts and the sequence in between the double-strand breaks is eliminated, and this is repaired. And we have also combined the 2 guide RNAs, but with a nickase, to create a staggered double-strand break. And this will potentially reduce the off-target effects, because you will need to have 2 nicks in close proximity in 2 off-target sites in order to have a effective double-strand break. So if we administer these vectors to mice, we see that the GO protein is reduced to almost or non-detectable levels. And if you look at the livers, this is with the nickase. The controls have a lot of GO expression in all the hepatocytes, while our treated mice show only some hepatocytes that still express GO. Okay, so After this, we decided also to explore what the modifications at the genome level were with these three strategies. So when we generate a double-strand break with a single guide RNA, what we see is that we have mostly small edits. For example, guide 1 tends more to insertion of 1 base pair. Guide 2 tends more to deletions of 1 or 2 base pairs, which I think it was defined by many others. Then when we do the 2 double-strand breaks, we mostly have the deletion of 60 base pairs, 64 in this case, which is exactly between the 2 double-strand breaks. And when we do the staggered double-strand break, what we see is that there is a very high variability of the edits. So we have insertions, deletions of many different sizes, many different sequences, and you see that the frequency of each of them is pretty low compared to the other ones. So this was interesting, and this could, tell you, for example, do you want to have this profile? Maybe not, but on the other end, you will have less of targets, as opposed to here that you have a very predictable on-target edit, but you have double the chances of off-target because you have 2 guide RNAs and a nuclease. But something we were curious about is that we had these insertions here that seemed to appear in all mice, and they were of unknown origin. So they weren't part of the genome of the mouse. So when we look at this, we see that the insertions that are higher than 20 nucleotides in the animals treated with the nuclease are mostly unknown. So they don't correspond to anything known in the genome. While for the nickase, only 5% of these insertions are of unknown origin. At this point, it was pretty obvious for us that this could be insertions of the AAV genome, which had already been described before, that when you generate a double-strand break, you make the AAV genome more prone to integrating in that site. So first thing we did was to generate a new pipeline to analyze indels and AAV insertions together. And using this pipeline, we saw that when we use the nickase, we have very little percentage of the indels are AAV integrations. While when we are working with a nuclease and we have a double-strand break, we see that the integration significantly increases in these animals. When we look at what is integrated, we see that mostly full-length or truncated ITRs are integrated. We have also worked, and I'm not showing this, with long-read sequencing, and we have seen that we have also full-length AAV integrations, and this result is probably biased because of the way we are doing the PCR and the sequencing. But we still know that we have high percentage of integration in these groups. Additionally, we also looked at the repair mechanisms. So we know that double-strand breaks preferentially are repaired by canonical non-homologous enjoining, while staggered double-strand breaks can use other pathways. So we looked at microhomologies in our data, and these microhomologies basically when you have a staggered double-strand break, and if there are homologous regions, but very, very small homologous regions in the 2 sites, they are repaired by microhomology. We saw that when we have staggered double-strand breaks with the nickase, we have a very high percentage of the indels are repaired by microhomology, while this is not the case for the nuclease Cas9. So this is also pretty interesting, and we are still working on understanding this better. So almost finishing, we also have looked at off-targets by sequencing in silico predicted off-targets by looking at circle-seq, cast-seq for large chromosomal rearrangements. We haven't found any off-target in any of our settings. This doesn't mean that we don't have off-targets. This just means that with the current methods, we are not able to find any. It might be because there is no off-targets, but this might be also a limitation of the technologies. And finally, just very briefly, we have also looked at therapeutic efficacy because it's our end goal. So we have tested, this is the nickase at different doses in a mouse model of primary hyperoxaluria, where we treat mice, and 4 weeks later, we challenge them with a precursor of oxalate. And when we look at oxalate in urine, we see that the treated animals have much less oxalate compared to the pH1 untreated control animals. And as previously mentioned, and as expected, we also see as an increase in glycolate that shouldn't be pathological. So with this, I get to the conclusions. I think the in vivo genome editing is a clinical reality right now. I think we have a decent amount of clinical trials already. Something I think you should bring home is delivery. We need to really think ahead of time in delivery before starting to decide our editing strategy. Each method has its pros and cons, and I think each delivery vector needs to be evaluated in the specific context. What is the target organ? Can we administer locally? Do we have to administer systemically? What type of CRISPR do we want to express? I think LNPs and AAVs currently are the most promising modalities for gene editing. And based on our experience, we have also seen that nucleases and nicases differ on how the edits are repaired, but they also influence on the integration of AAV genomes, which is something that we should keep in mind as well. And if you have any questions or if you want to contact me, here is my email. And the most important slide, I want to thank everyone, well, especially Gloria, Laura, and Martin. Yes, it's Laura, the person that is chairing this. Laura was our PhD student, and now she has moved on and she's doing a postdoc. But the work that I've shown in primary hyperoxaluria was started during actually my PhD, and all the nickase part was developed by Laura during her PhD, and that's our most recent data. I want to thank the collaborators, and also I haven't shown any data from my Boston lab, but I want to also thank everyone in the Boston lab because they have, like their work is amazing as well. So thank you. I'll be happy to take questions, Laura.
Thank you, Nerea. Thanks. It was an incredible presentation, and not just because it was most of our work, but because it's always amazing listening to you, and especially because your educational overview was incredible. And since we know that our main targets are indeed young researchers, it's always nice to have this massive view on AVs especially. Thanks also for getting to LNPs, which was one of my questions at the beginning. Can we compare these two systems? Which is the CRISPR-Med just ended, and we had the amazing presentation, also the CRISPR-Meds. And in between these different deliveries was again one of the most discussed topics, as well as safety. And so thanks for getting to these different approaches. So I've seen a lot of hands up during your presentation, so I believe the audience would like to ask you some questions. And while I wait for a question in the Q&A, I want to just remind the audience that please type your question in the Q&A, and so then I can proceed reading these questions to Nerea. I also have some questions, so I can start and break the ice. Between the different part of the presentation that you mentioned, there was some stuff on AVs, well, on the basic AV sides, and some other on AVs and genome editing. In terms of basic AV, there was, well, basic AV, all this, you mentioned at some point that there is something that we still don't know in terms of tropisms of AVs. And you said that tropism depends on the capsid, and just probably something on capsid they make an AVs specific for one tissue and not other. But we also know that some AVs is able to transduce multiple tissues. And so I was wondering if there is something in common between these different tissues and why this AV is capable of entering. Is this due to receptor that maybe is AVs as a bit more on this, so for our audience to understand better?
Yeah, that's an interesting question. And I think they have common receptors. So for example, we know that, and it has been named AAV receptor. So this is a protein that is ubiquitous. It's in all the cells, and it's essential for most AAV capsids to transduce the cell. But this is ubiquitous, so this is not telling us much. We also have other attachment factors such as sialic acid, galactose, but these are also pretty ubiquitous, right? So this is not telling us as much. We also know that there are other, like proteins that are involved in the cellular trafficking of AAV. But I think something we should keep in mind, for example, AAV9, right? AAV9 transduces the liver very well, for example, in a mouse, the liver, heart very well, and it also transduces CNS. But this is probably because AAV9 has different capabilities. So it transduces the liver because everything transduces the liver. It's very accessible. It transduces the brain because it can cross the blood-brain barrier. So AAV9 probably has a capability to cross the blood-brain barrier, and then it transduces the brain. If you were to inject an AAV directly in the brain, for example, AAV2, it transduces the brain, but not systemically. So it's not only about what the cell has, but it's also about what physiological barriers the AAV encounters. And it's probably the same for the heart. So the heart, we know that the vessels in the heart are, continuous endothelium, so it's very difficult to penetrate that endothelium. So AAV9 might have something that allows it to cross. So it's not only the cell, the target cell, it's also the physiological context. So this is very complex, and it might not transduce pancreas, but maybe it's because in pancreas there is some intracellular trafficking that it's not happening. So, it's many, many different factors, not only the surface receptors of the.
Okay, thank you so much. And then I was wondering also, because at the beginning you said, okay, these two mains are AVs from one side and LMPs. And with AVs, we reach different tissues. With LMPs, we are still not at the same level, but it's also most likely true that LMPs in terms of genome editing, for example, is a new and a super new and hot topic. So it's something that we don't have so much understanding on, most likely, and we have so much to improve on LMPs to make it better. And I was wondering, and we learn a lot in CRISPR-Med, and there's massive groups which are working early on LMPs, and they reach different tissues which are not only, and they have been able to target from liver. But one of tissue that is still not reachable, as far as understood, or two actually, muscle from one side and kidneys. Do you have any clue why it's so difficult, for example, to target kidneys? And because I know that you are, and we presented at the beginning that you will base your research on kidneys. So why working with AVs on kidneys and not LMPs, for example?
Yeah. So as I said, I can't speak much about LMPs because it's not my field of expertise. So for me, it's at this point difficult to know why LMPs wouldn't be able to transduce the kidney. If we inject them systemically, if you think about the function of the kidney aims to very specifically select what goes to the kidney parenchyma, so what goes to urine. So, if you want to get to kidney parenchyma, it's going to be very difficult to go through the blood because the blood is going to be completely filtered. So the kidney wants to avoid at all cost to lose proteins in urine. So if you inject an AAV systemically, it is a protein, the kidney is gonna be like, this is not coming in. So, I think it's the structure and the function of the kidney that makes it so difficult. We are working on administering it locally by different techniques, and this shows to be, and this is published as well, this is better, at least with an AAV. I'm not sure with LMPs though. Yeah, there is a size limitation as well, because you have to go through the glomerulus, which has a very small size. So yeah, I think it's the type of tissue, like liver, for example. The goal of the liver is to screen everything that is in the blood, right? Like everything that comes in, the liver is gonna be like, okay, let's see what this is. Let's see if I need to metabolize it. So that's why a liver is much more prepared to accept your vector.
And then, since you mentioned now that you are considering like in local strategies instead of intravenous, and so could be this possible to translate into the clinic, and which can be the main concern in terms of toxicity for this translation of AAVs to the clinic eventually?
So I think, we know that there have been many instances where we have seen AAV toxicity in the clinic. So, for example, we know that there is a certain dose that we cannot go above it, because that's going to cause liver toxicity. It's going to cause activation of complement and all kinds of issues. For example, transduction of DRGs, which is also something that has been shown and that can cause also some central nervous system, peripheral nervous system toxicity. So yes, I think the biggest issue is the dose. So if we were able to have AAVs that are efficient enough, reaching and specific enough to reach your target tissue, you would avoid most of these toxicities. There is always going to be some immune activation, right? Like we are introducing a viral capsid in the system is going to detect it. But it doesn't mean that that's going to be necessarily toxic.
And then maybe if, since our focus has been genome editing, so I was wondering, one of the other limitations that you mentioned for using AAVs as delivery system is the fact that you will have Cas9 or any other of your editors persistently express. Do you know if is there anybody who is working on optimizing these constructs to limit the expression of Cas9 and to prevent this permanent expression of Cas9, which can also result in immunogenicity, as you said?
Yeah, so there are several approaches. Traditionally, people have worked a lot with inducible promoters. This has been studied for many, many years. The problem of inducible promoters is that they are leaky. So even though you can increase the expression upon induction, they still leak a little bit. So that might not solve the immunogenicity issue. It might solve maybe the target issue, I don't know. But there are some other new strategies. For example, there are these introns that can be specifically removed. With a drug, so if you don't have the drug, there is no translation of your RNA. If there is the drug, you have the splicings and you have the translation. Some people have used degrons as well. With that, you won't have protein, but you still have antigen from the protein, so you could still have immune responses. Yeah, that's I think mostly the strategies that people have used. Well, some people have used also siRNA targets, not in the CRISPR context, but for example, you can put like a 3' UTR to your transgene that has a target for a microRNA, and reduce the expression, for example, in the liver. But yeah, right now we are not able to achieve a transient expression with AAV as we would with an LNP.
Thank you. And then it was something that came to my mind also. I know that other peoples have been using also strategy, which is called like Kamikaze9, which what this Kamikaze9 does is cutting the same Cas9. And so this way you are blocking basically the expression of Cas. Of course, using this inducible promoter as well, you can use it to block your expression of Cas once Cas has done what you were expected to have. But then I was wondering, you show us that there's also a possibility of having integration of your AAV. So with this strategy, potentially, since you're cutting your AAVs, you can also increase eventually the possibility of having integration of small fragments. Do you know anything about this?
Yeah,
Or
that's actually a really great point, because you know, the AAV that is integrated is usually the AAV that is probably not in a concatamer. So AAV genomes, when they are in the cell nucleus, they stabilize themselves by forming these concatamers, which are AAV genomes that are bound together, ITR to ITR, and they are very, it's like a huge plasmid, let's say, and they are very stable. So if you go and cut the concatamers, you might have a higher chance for integration. That's actually a very interesting question, and we could look into it as well.
Yeah, because I was wondering what you presented was mainly integration through ITRs.
Yeah.
But yeah, could be also be the case for any other fragments that can be captured also through these broken hands mediated by Cas. So with this, I think I can go ahead and with all my questions and also audience too. But we have your contacts and you share with the audience. So this means that they can reach you out and ask you any of the potential question or ask you more details also on your work. With this, we reached the end of our webinar for today. I want to thank you for the amazing presentation. Thanks also for sharing part of the work we have been done together. And then thanks to the audience for attending this webinar. Thanks to Geneomdi for supporting us always, and especially supporting young researchers, Cost Action, and CRISPR Medicine News for giving us the possibility to make these all these presentations visible. And especially want to remember everybody that this is a record sessions. This means that you can always have access to the presentation on demand. So thank you, Nerea, once again, and I'll see you all the audience very soon with a next presentation, most likely on delivery or as well on safety. Thanks a lot.
Yeah.